Right. So we have some excitement brewing here for the talk. That's good to see. Thank you for the introduction, and it's really wonderful to be here to talk a bit about our research. I'm going to spend a little bit of time talking about first the natural history study, what that is. Then I'm going to talk about something very recent, in fact, a publication that just came out about a week or two ago on zebrafish. So this is going to take us into genetics, and then we're going to come back to gene therapy in mice. So that's the overview of the talk today. Well, I usually always want to start off by acknowledging the great team that I've been privileged to work with at the NIH. For those who don't know, I'm going to show a picture of what the NIH looks like in a minute. But believe it or not, this scene here is actually pretty close to the NIH. It's some of the most advanced whitewater in the United States, the Potomac River. And many, many years ago, for those who are interested in history, there was great difficulties to try to get goods and services from Washington, D.C. into the Great Lake area of the United States, because you had to go through this. Now it's a whitewater mecca, and it's about 5 miles from NIH. This is my research group. We have a very large and diverse group with people that are real experts on clinical medicine, including Dr. Manoli and Dr. Gallaretta, and Dr. Oleg Shalachkov, who started the PA protocol in 2016 at NIH. We have a number of people in the laboratory that have been working really hard for many years on gene therapy, including my colleague Randy Chandler, who actually developed a number of gene therapeutics, mouse models for PA, and continues to work actively in the space. Some people know Jennifer Sloan, who's in my group. She's a genetic counselor, also has a PhD, and she has expertise in animal modeling as well. And we have lots of other people here that are students over the years that have joined the group. They typically stay, and then they move on to medical or graduate school. And then our most recent staff member is Dr. Carolina Gallaretta, who some of you might know. She's an expert on PA and is a clinical and biochemical geneticist. So that's our team, and they all have been contributing to the work that I'm going to talk about today. I do have some disclosures right now. There's nothing related to PA. I do have a number of patents. The NIH holds them, and I don't think any are in conflict with today's presentation, but those are them if you want to look them up. I mentioned what we're going to cover today, and I'm going to start off talking a bit about what a natural history study is. And I'll give you one example of what you can learn from a natural history study. Before I do that, I'll say just the cursory words about propionic acidemia to sort of set the stage, maybe so other people won't have to talk about this. Propionic acidemia is part of a group of disorders of propionyl-CoA oxidation, and there's many of them. It happens to be the one that was the first one described by Barton Childs in 1961. The index case had propionic acidemia. For those who are really interested, that paper is an absolute must-read. That paper came out in 1961. The studies began on that patient in 1959. So most patients have, if they have PA, they have massive excretion of PA, or if they have MMA, it's methylmalonic acidemia. And in the early days of this disorder, it wasn't known if a patient had MMA or PA. They had something called hyperglycinemia, ketotic hyperglycinemia. So they had high ketone bodies and glycine, high levels of glycine in the urine and the blood. And it wasn't until the late 60s it was realized that that's a group of disorders, organic acidemias, and PA is one of them. Those are what cause most of the patients to have a problem. It's either mutase deficiency or problems with propionyl-CoA carboxylase. That causes propionic acidemia. And as many of the families here know, this is a very difficult disorder to treat, and it has a guarded prognosis. And it's prototypical of a group of disorders called organic acidemias, of which there are hundreds. So one of the ways that the field should think of this is, if we can figure it out and solve it for PA and MMA, we can extend it to other disorders that might be less common. Maybe they're going to be more severe, but there's going to be lessons learned for everybody. Okay, now we're going to start getting heavy into pathways and such. So I'm going to try to walk everyone through this, and I realize it's going to be a little complicated. What I'm showing you here is a little sac over here. This is a mitochondria. So inside our cells, there's these energy factories called mitochondria. And the enzyme that causes propionic acidemia is inside of this enzyme factory, the mitochondria. And that's what this is. These little things are what we call cristae. And what I'm showing you here are the two main genes that cause propionic acidemia when they have mutations in them or changes, PCC-A and PCC-B, or alpha and beta. This enzyme requires something called biotin, and that's a separate discussion. But if a patient has propionic acidemia, depending on where you are from, you either can have the A problem or the B problem. Many of the people in the Amish and Plain community have the B problem. But if we look at a larger group of patients around the whole world, A and B are 50-50. Now, PA comes from a problem metabolizing propionyl-CoA, and that comes from branched-chain amino acids mainly. And that's valine and isoleucine. So that's why for those who are in the audience whose children have this, we restrict protein. The reason we're doing that is to reduce valine and isoleucine to give just enough to grow, but not too much. Now, propionate also comes from the gut, from the bacteria that live inside our intestines. Maybe about 30% of it comes from that source. So we're always making it in our bodies. And that therein is the problem of this condition. You can't just stop the metabolite generation. What ends up happening after you make propionyl-CoA, it turns into the Krebs cycle intermediate succinyl-CoA, and that's where the MUT reaction is, the MMA mutase reaction. So the reason why these conditions are grouped together is they're very, very closely related biochemically. Now, if someone has PA, there are certain things we look for in their blood and urine, biomarkers. Things like 2-methylcitrate, 3-hydroxypropionate, propionyl carnitine, which we detect in the newborn blood spot. And then there's other biomarkers as well. Those are the ones that are most commonly used in blood. At the NIH, there's something else that we do that maybe some of the patients here have done, which is we do a carbon-13 propionate oxidation whole body study where a person drinks this non-radioactive form of propionate, and if they have the enzyme active in their liver, they release carbon-13 gas in their breath. So they take this drink, blow out, and you can tell how severe someone is affected by this method. It's not offered everywhere. Now, what do we do for the patients? The number one thing we've been doing, and by the way, this has been done, the very first patient was treated in 1959 and we reported in 1961. That was this, we're doing the same things. We're restricting protein. Carnitine supplementation is used, and there's a separate discussion about this, and there's a reason for this. I think it does help, but there's a debate on the mechanism and the potential to overdo this. People can take a lot of carnitine. You don't always absorb it, and there's other comments that I'm not going to make about carnitine, but it's a useful adjunct for the patients. It's not a cure. Sometimes we give people antibiotics. Why do you do that? Because that sterilizes the gut and reduces the flow of propionate from the gut into the liver, where it then turns into the metabolites. That's why you take antibiotics. Some people think they benefit from it, others don't. Then there's other supportive care measures that we use when someone has a problem with their organs. You have to treat the organ dysfunction. Now, what some people would consider as a definitive treatment for this, if a person has a severe form of it, is a liver and/or liver kidney transplant, and/or in some patients, heart transplants, and/or liver heart transplants. These are very, very aggressive treatments. It's not really a treatment, it's a surgical procedure. And then there's a range of new therapies. I'm calling them genomic therapies because I include mRNA editing and AAV and also oligonucleotide therapeutics in this category. And none are approved for patient use. Okay. So if a person has PA, I'm trying to get into the idea of a natural history study. Not everybody, if you look at this little cartoon of this little person over here, you can see some of the things that might happen if someone has PA. For example, they might have hyperammonemia, but not all patients get that. They may have metabolic acidosis. Not everyone gets that either. They may have intellectual impairment. Not everyone has that. They may develop a stroke. Not everyone gets that, etc. So there are some symptoms and signs that are more common in the patients. For example, there's going to be discussions here on cardiomyopathy. Not all patients get it. Some patients get it earlier than others. We still don't understand the mechanism of this. We don't have any good animal models where we could take a mouse or other animal and say, that mouse is going to get the same thing the patient gets. We're not there yet. With that comes risks of arrhythmias and other problems related to heart. That's going to be the topic of lots of other discussions today. We're recognizing that chronic kidney disease is part of this condition for some patients. Again, not every patient, but some patients develop this. Why they develop it, what's the mechanism, what's the biomarkers, that's unknown, etc. You can see here all these symptoms and signs. Not everyone gets every symptom and sign. And that's part of the challenge of management, to figure out who's going to get what and what treatment you could do to stop one of these things from coming on in a patient. Many patients have metabolic instability. And what this means is if they develop a common virus, runny nose, cold syndrome, they can get very, very sick, literally have hyperammonemia and encephalopathy. It can be quick, but not everybody has the same degree of instability. Kids that have this, especially problems with high ammonia, those are the children, and I've seen infants offered liver transplants, elective. In PA, the liver is not intrinsically damaged that it has liver failure. The reason you do a liver transplant is to stop the instability, and there's specific reasons for that, again, which I would say are not fully understood, but we know that when someone gets a liver transplant, they don't have instability anymore. Pretty aggressive treatment, quote-unquote treatment. If someone has very advanced kidney disease, they develop something called chronic kidney disease that progresses. And the end of the treatment of this is renal replacement therapy and/or kidney transplantation. And if someone has a severe heart problem that leads to heart failure, they could require a heart transplant, or they could require a combination of these things like a liver heart transplant. So these are very, very aggressive treatments. Not every patient needs everything. And that's part of the challenge, to figure out when you see families and meet patients who needs what and why, and when should you do it. I just mentioned the disease management approaches, but to say that no matter what these are, none of them will stop all the symptoms and signs in the patients. So how do you study this condition? So what we do is we use clinical research, and thank you very much for the kind comments that were offered. We've seen many, many patients, hundreds and hundreds of patients over the years with MMA and PA syndromes. Then we use our learnings from that to make animal models, whether they're cell models. We started off using worms, C. elegans models, mouse models, and I'm going to talk to you today about zebrafish. So this is going to be, just get ready for this one. It's interesting, but it's going to be, it might be harder to fully grasp. And then we could go back and forth. So if we discover something in a patient, then we can go to the mice or fish. If we discover something in an animal, we can go back to the patients and see, do they have it? And we've done this many, many times. We've written lots and lots of papers to show that this approach is really, really powerful, especially for finding biomarkers and testing new therapies. So to talk a bit about a natural history study, I set it up for you to just try to explain the difficulties of clinical management and trying to ascertain in any patient what is going to happen to them. Parents want to know, and the patients want to know, what's going to happen to my child? What's going to happen to me? And this is part of how we can address this, a natural history study. And this is conducted at the National Institutes of Health, which still exists. It's in Bethesda, Maryland. It's the largest clinical research center in the world. It absolutely dwarfs anything else anywhere. We have 200 dedicated inpatient beds. That means every patient is there for research. This is an old number. We have about 100,000 outpatient visits per year, and there's many, many patients with rare and difficult to treat conditions. That's what this entire institution is focused on. And it's part of the federal government budget. And I work for the federal government. And I'm here with official approval, by the way, of the federal government to talk today. A natural history study requires teamwork. It requires families to participate. It requires the coordinating center, that's us. It requires local physicians to all work together to try to obtain information that's going to help us determine what is the natural outcome, the prognosis of a condition, when there's no intervention applied a priori. And so what we do is admit patients, and I know families have been to the NIH. Usually they come on a weekend, they'll stay overnight, we'll pay for the accommodations, and then they undergo what I usually like to say is a battery of tests that would take, in most places, it would take many months to a year to get done all the things we can do in a few months, in a week. We have a cadre of experts that have seen many, many patients, so we have specific evaluations, and you can see some of the things that we do in our study. At the end of it, it's about 500 different variables, including biomarkers, imaging studies, and metabolic measurements that we all are contained in a database at the NIH as part of the clinical record. It takes about 2 to 5 days, and sometimes patients have to come back because we can't finish everything. If some detailed scans are needed, they may have to come back. This is how many patients we've seen, and the power of this is allows you to see many patients over a long period of time. We've evaluated over 60 patients with PA, and so some of the things you can do from a natural history study is to say, who gets what treatment? Now, this is not prospective in terms of capturing an entire population, but it gives you an idea that if you look at 62 patients in our cohort, 8 had liver transplants. 1 had a liver heart transplant. 1 had a liver kidney transplant. 1 had a kidney-only transplant. And then 52 did not have transplants. So you can do something else. You can even go further and say, who got what? Is there a difference between who gets the transplant and not? What are the biomarkers? We can do all those things. And you can see this is not fully updated, but alpha versus beta, who gets a transplant, it doesn't, to me, look like there's any difference. So in other words, if you're beta defect and you have a severe problem, you have this equal chance of getting a transplant if you're alpha type. For example, that's just one of the things you could do. And that's very descriptive. Now I'm going to give you an example that I've actually talked about before, but I think it's important for the families to know this. There's something else you can do, which is when you put together lots of patients that have different treatment approaches, different genotypes, you can examine things in an unbiased perspective. For example, kidney disease. It used to be thought that PA patients maybe developed kidney disease. People weren't certain about it. It's quite common in the patients. And here's an example of how we were looking at this. And there's a specific reason you want to know about kidney function. It's very, very important for clinical trials and for treatment. Many medicines have something called nephrotoxicity. So if you are a person that has CKD, you should avoid medicines that can damage the kidney. But not everyone, if you don't know, you might not do that. For example, so here's this is, by the way, Oleg Shlachkov prepared this. I think it's wonderful. So here's just a number of different participants. We take all their values and bring them in together, and then all of a sudden what you do is if you aggregate them, you can find out there's a trend. And here's the trend. Kidney function goes down with age. This is what happens with CKD. And when this happens, there can be an effect on cardiac function. You must know this if you're doing anything else for a patient if you want to study systemic manifestations. And so this, by the way, is in a publication. You can access it. I'm going to give you the PMID in a minute. But this is just some of the reasons why this really matters. If a patient has kidney disease, when should you screen for it? How should you screen for it? How do you manage it? What medicines should you take? What's the trajectory of the change, and how's that going to influence other things in the person's body, like their heart, or their eyes, or their nerves? Could we make drugs that target those? Are certain drugs better? And then this is way down the road, but if someone wants to make a prediction when the filtration rate is going to go to a critical level, and when it's critically low, a person needs renal replacement therapy. That's dialysis or transplant. This is very important to understand. And I talked to you about medicine doses. And there's another reason. Obviously, part of what is exciting, I think, is if someone is going to have a new therapeutic, this is very important to understand. If you don't have a really good read on your renal function going into a study, and then you get reassessed later and someone says, wow, there's this renal function, there's an effect on the kidney, and you don't know about that a priori, you could think that your medicine is causing kidney damage. It might not. Or alternatively, it might be causing damage and you didn't figure it out because you didn't monitor correctly for it. So it's very, very important. All right, now that we warmed up, this is the time for everyone to take a breath in, because we're going to start talking about animal modeling. So we're going to move into genetics to try to figure out how do you really figure this out when you can just do anything you want and do a thought experiment. How would you figure it out? One thing you can do is use animals. There's a reason you do that, because if the animals have the condition, you can manipulate them humanely and under the law. And we do that. We take great care of our mice and fish. So we're doing this with mice. We did it before with worms, tiny little worms called Cenorhabditis elegans. It turns out they have genes that are very similar for PA and MMA as well. Mice, of course, are the old standby. But I'm going to talk today about zebrafish modeling. So I'm going to show you pictures of zebrafish, but I want you to know, if you haven't seen zebrafish, a full-grown zebrafish is really about the size of a minnow. It's not some big, giant striped bass like in a pond or something like that. It's a tiny little thing. So the pictures will make it look like you're ready to, have a trophy for your home on display, but they're not. They're tiny little minnows. So why would you use a zebrafish? Well, you can see here. Here's a schematic of a little person. You can see their brain. This is supposed to be their brain. There's their heart, liver, kidneys, etc. It turns out zebrafish have all those things. They have hearts, they have brains, they have eyes, they have incredible visual capacities because they need really great vision to find the little things in the water and eat them. They have hearts. And when they're very little, they're actually transparent. So you can just visualize them and look at the heartbeat. You don't have to do anything to the animal. You just put it down and look at it in a microscope. So there's incredible power to use these model systems. And I want to tell you today about a paper we just published. I'm going to give you the highest level assessment of this. It just came out. It's called Metabolic Rerouting of Valine and Isoleucine Oxidation Increases Survival, and you can read the rest of the title. It's quite a technical paper. It was led by a great colleague of mine in the laboratory, Sung-Kook Hong, who's a staff scientist, who's a zebrafish genetics expert. And I want to acknowledge that some of this work was funded by the PAF from a pilot grant, and I'm very, very appreciative, as my lab is, for the support. Going back to our pathway, remember, there's the problem of PA. And what we're going to try to do with this, I'm going to try to keep reiterating this, is if you have a pathway and it's going A, B, C, D, let's just imagine that you're going A to D. And the problem is from C to D. Well, one thing you could do is block from A to B. Because if B was going to turn into something bad at the end, say D, which is propionyl-CoA, maybe if you reduce that, the effects of a problem going from C to D in the pathway would be less. And that's what I'm showing you here. So this would be like C to D, the step of the pathway. We want to try to reduce flux through this pathway. And what you can do is block something upstream. And you want to block something that when you block it doesn't cause a problem. And it turns out humans that have problems with this gene or this gene have metabolic elevations in their blood and urine, but don't have a clinical syndrome. So the theory would be to block one of these or both and convert a fish now with propionic acidemia into one of these milder forms of a mild condition that has humans it's asymptomatic other than biochemistry. So can this be done? How would you do this? This is where you use genetics. And this is, I'm not going to have a time to really explain how we do this, but going back to our fish, one of the first things you can do is you can look at a database and say, in silico, in the computer, do the genes exist? The same genes that humans have, do they exist in the fish? Answer, yes. And I'm showing you the information on the other side of the screen that shows this, and there's other ways you can prove this. But those similarities and identities are massive. And you can complement genes. So you could take a gene from a fish, correct a human cell, take a gene from a human, correct a fish. That's how similar these genes are. It also means if you made a model, there's something else about zebrafish that, as an aside for experts, sometimes they have multiple copies of a gene. And that's not good for modeling. And zebrafish have one copy, single copy. So that's very good for us. So what you can do now is use genetic engineering. I'm not going to explain how this works other than to say there's ways if you know the DNA code, you can change that code and eliminate the gene. And there's different ways to do that. We did that. You can eliminate the PCC-A gene, B gene, and then you can see what happens to the fish. How do you do this? You use zebrafish genetics. You breed the fish together. There's a room this big full of fish tanks. Not just from my projects, but from others. So it's quite a big operation to find these little fish. And then what you do is breed them, then you take a tiny portion of their fin and you use that little bit of their fin to do a genetic identification to see if that's the one that has it. So that tells you if it worked or not. Then you can do things like imaging and see how the fish do. You can check their behavior. You can examine their development. And then what you can do is see if they make the metabolites. Do they have the metabolites? And so the way you do that, and by the way, these are just some of the metrics you can use, and for those who are really interested, you could look this up and read about it if you want. You can use biochemical assays to see if they make the metabolites using acylcarnitine ester profile and index metabolite measurements. So you can measure to see if, does it produce the signature of PA and MMA? And of course, you can see what the phenotype is. So that's the approach. All right, then what you're going to do next is now you're going to make a double and triple mutant. I should say we did this. And what is done now is going after those genes I told you about a minute ago, and you block those. And now you breed them to the other fish that have propionyl-CoA carboxylase mutations. And you make double and triple mutants. So fish that are missing the PCC-A or B gene, and one of these or both of those. This is very hard to do. And that's how you do it, and it is a specific way to do this, and those are the defects, and those are the metabolites. Patients that have a defect let me just give you an example of an enzyme called isobutyryl-CoA dehydrogenase deficiency make something called isobutyrylglycine and isobutyrylcarnitine. So if you make a fish mutant that's missing this gene, it better have those metabolites. It does. You can read the paper if you want to find out the excruciating details, which were performed, by the way, by Lisa Kratz, who's the director of the Kennedy Krieger lab at Johns Hopkins University. So one of the things we learned about this is part of when you do research, you don't really always know what's going to happen, right? When you make the single mutant, so going back, I'm going to try to walk people through this. If you just take one of these out or one of those out, that's a single mutant. And then if you take both out, that's a double mutant. So the single mutants look normal, which is what you would, based on human genetics, tells us that. The patients that have the single mutation are normal. But when we make the double mutant, and by the way, this is not to scale, but this double mutant, this little guy down here, this is a fish that's been engineered to miss both those genes. It's very small, and it dies. So that tells us that these pathways in humans have some degree of redundancy, but it's not absolute. Because if it was absolute, the double mutant would be normal. And you can do survival statistics, which we did in here, and I'm showing you that on day 60, the double mutant, that's the green. The way you do these analyses is you go to the fish tank at day 30, 45, 60, you just take all the fish out, euthanize them, and figure out who's alive. They all die. Now we're getting to the actual study, which is now we're going to combine the beta defect mutant with our upstream mutants to make double and triple mutants, and ask the question, does it affect survival? Okay, and the answer is it does. So if you make the single mutant, they always die by day, and there's different types of survival analysis one does with this. With fish that have the single or double mutation live longer. Now what was unexpected, we thought that the PCCB mutant, when we made it a triple mutant, in other words, missing PCCB and then both the other genes, would live as long as this mutant, and it doesn't. And there's a reason for that, because the metabolites are coming from other places than branched-chain amino acid oxidation. Nevertheless, it's quite important, because this is a very, very strict model. This is a model that uses lethality. And you see there's effects of lethality. This means that if you dialed it down, it might have, it would be promising to put into a different approach that's not as extreme as making a full knockout. So I know this is really hard for people to grasp. I'm just trying to get through this for the group. So there's things you can do from this, and this is not a new idea that we came up with, but it really shows that it probably could work. You could use inhibitors of these molecules, these other enzymes, small molecule inhibitors. That might help PA. It's not going to affect the PA enzyme, but it's going to stop flux. And I think one of our speakers is going to talk about flux soon. And then it speaks to another approach that one can use, which is more advanced, about how you could potentially target multiple ways, multiple genes that are below early steps in branched-chain amino acid oxidation. Could you get the same effect, or could you synergize? And so there's ways you can do this using mutagenesis, animal mutants, and then also now in vivo somatic gene editing in mice. Okay, I think I have a few more minutes to finish up at the end here about gene therapy. And so this, again, I've talked about this before to the group, but if people don't know what a gene therapy it is, more broadly defined, it's a transfer of genetic material into the patient or their cells. And what you usually do with gene therapy is you introduce the gene back. So if the gene's missing or damaged, you put a copy back. That's called gene addition therapy. You can also try to edit the gene, change the DNA in the genome. And I'm going to talk a little bit about that at the end, but people may have heard about baby KJ. It's been on the news. That's gene editing. So there's a vocabulary to know about. When I talk about a vector, what I mean is what's carrying the gene. The transgene is what's inside the vector. In this case, it would be an AAV vector, the one I'm going to talk about. And then when we talk about transduction, this is how when the virus infects a cell. So you need all these things to get it to work. You need a vector, you need a transgene, and you need to get transduction. And there's things we would want in an ideal gene therapy product. And by the way, nothing is ideal. No medicine is ideal. Everything has toxic effects, and gene therapeutics are no different. But if we could design one de novo in a thought experiment, we would say we want it to be delivered by injection. We could control it. It would target to where we want in PA, that's the liver and the heart. It would be stable, and it wouldn't have any toxicity. Nothing will reach that level of criteria, but that's what we would want. That's ideal. And what we are using to do this, as others, there's hundreds and hundreds of people doing this type of a virus, is using something called adeno-associated virus. It's actually a dependovirus, which means in itself, it can't replicate on its own. It needs what's called a helper virus. So over the years, about 50 years, it's taken to take adeno-associated virus and convert it into a gene therapy vector. Now, what's super exciting about this is that you can reprogram the vector. You can change where it goes, liver, heart, muscle, eye, brain. And if it works, in some animals, a single injection can produce expression of the gene for the lifetime of the animal. So this is really important for gene therapeutics, for patients, right? You could potentially get one treatment and that would be it. That's what everyone wants, whether we're going to get there. In animals, it works. This has been used in human trials, and there's 7 FDA-approved indications for other types of AAVs for human use. So it's one thing. You're going to hear about RNA therapy, that's something else, and there's even other things. So what we're doing is, this is the idea of this project that is funded by the government, called Platform Vector Gene Therapy, and another one that's a government philanthropic corporate enterprise called PAVE-GT, and that's at the bottom. Bespoke Gene Therapeutic, BGCT. PAVE uses PCC-A, the Bespoke is PCC-B. I'm only going to talk about A today. But this is the idea in schematic form for those who would want to think about it. You would put this vector into an IV, into a fluid, and you would give the patient an IV infusion. It would go into their body, and then it will go to the liver. All AAVs hemoconcentrate, or hepatoconcentrate. It would concentrate in their liver, and also, depending on the serotype and dose, will go to the heart. That is key. Those are the organs that tell cells that must be corrected in this disorder. So with the Platform Vector Gene Therapy Project, I want to just mention this one because it's a project we're working on with NCATS, which is a different group at the NIH. And this tries to develop a platform approach. So in other words, a person could come after this project and say, I want to work on glutaric aciduria type 1. Let me take your example, and I'm just going to change the gene. Or methylglutaconic aciduria, for example. I'm using organic acidemias. And you change the gene out. All of our information is being disseminated into the public domain, which means anyone can access it. It's a fairly large team. And you can see my mugshot, there I am, right there. But NCATS is leading the effort in terms of supporting this project. There's a group in the Genome Research Institute that I lead, and then there's another one in the Neurologic Institute that's working on neurologic indications that Dr. Karsten Poneman leads. So there's a lot of us working on this. And right now, for the alpha defect, we're here. We're ready to submit an IND, so we've actually developed the vector, tested it in mice, manufactured it. And we're going to start, the next thing is to manufacture and give it to patients. In a trial. In a trial. It won't be available by prescription. And then disseminate that information. So if anybody else wants to copy us And this is the big difference between what we're doing in a company. We're going to let other people copy us because you all, through government taxes, are helping support the effort. So this is going to advance the field greatly. And we've written a number of papers on this, and if anybody wants to download them, they're technical documents. These tend to be white papers about describing how you do it and what are the steps and how you interact with FDA. And you could access our materials right here. Just take that barcode or go onto the computer and type those things in, and you can get them freely available. Now I'm going to finish with just showing you proof of concept in mice and how we did it. So we used the PCC-A mice. We made them. These are very severe mice. They have a deletion of the gene, and they make no enzyme. This is the most extreme phenotype. And most people that have published papers in the PA space do not use these mice because they're too hard to work with, and most people can't get things to work in them. That's why. But they're very good to see if you have a really potent therapeutic, can it work? Yes or no, because you can use a signal, live or die. And then you can look at biomarkers, expression, transgene transduction, RNA levels, and enzyme assays. And we've done all this. And the goal is to target the liver and the heart. First thing you do is a dose-response study. So you take the vector and give it to mice. And what I'm showing you here is a survival curve. This is why you need to use a severely affected animal model. It recapitulates the most severe end of the phenotypic spectrum for patients. And unfortunately, many patients, if they're not treated aggressively and early, succumb in the neonatal period. It's very, very sad, and unfortunately, that still happens. And that's what happens to the mice. When they don't have anything, this means dead. They die by 2 days of life. If we use a low dose of the virus, it doesn't work. If we use 10 times more virus, you can see what's happening. They're living to weaning, then a little bit after, then they die. And if we use even more virus, they do quite well and live a long time. Dose response. Now, this is not analogous necessarily to a human, because neonatal knockout mice are very sick and they're small. That's not the first target for a human trial. But if it works in that model, it will work in the human setting in a larger animal model, because this is extreme, severe animal model. And we have approval for this, by the way. So then what you can do is say, did my gene get there? Did it get to the liver and to the heart? Answer, yes. Here's a picture. These little brown dots are cells that are expressing the gene in the liver. And this is on day 60. And you can see in the heart, there's even more, and the dose goes, as you go up, you get more expression. And then you can do something else, again with mice. You can take the organs out and measure how much you got in there. And what you're seeing here in these mice, that's the liver, this little green band over here, that's the enzyme. It's in the liver of the mice. And look, there's plenty of it in the heart. Now, this is different than any other types of therapies that might be out there, because this goes to liver and heart. And what we know from evolving patient data is that if someone gets a liver transplant, it doesn't fully protect against cardiomyopathy. There's been multiple reports of this, and we've seen it in our clinical study. So you really, ideally, would want to target the heart if it's possible, and this does. And now I'm going to finish with just a commentary about, maybe people have heard about this baby KJ who had this very serious metabolic disorder, not that PA is not, called carbamyl phosphate synthetase deficiency, which this disorder is so serious that the patient basically was living in the hospital his whole life until he could get a liver transplant. And he got a CRISPR therapeutic. And so there's been a lot of interest in this, because I can say myself, maybe other metabolic physicians in the audience, everyone wants to see if can my child get that. And so these are just some comparisons. The baby KJ, he got a CRISPR treatment for the liver. His disorder is mainly in the liver, and it's just for his mutation. Whereas an AAV therapeutic, and other ones that you might hear about today, they will work on patients that have all the different types of gene changes. And the AAVs, at least the ones we're working on, there's even other things that one could say about this, target liver and heart. In this AAV, I didn't show you, it actually goes into the skeletal muscle, and at high doses it goes into the brain. And this is our project is a scientific collaboration, and this is a large consortium that NIH funded part of this project too, by the way. So just some people get interested, and that's why I'm putting it in there. Not that it's directly relevant to propionic acidemia, but people always ask about that. And then just to say we've done the same thing for the beta defect, which I'm not going to talk about, but you can see here we've made multiple new mouse models of that disorder and have multiple successful gene therapeutics for that as well. So I will conclude by telling you that we have developed a number of potent AAV gene addition therapies for PA as well as other organic acidemias. They are experimental when they go to the clinic. You can't get a prescription for this. It could be a one-time treatment. That's what we hope, but we don't know. You always need to test the therapies, and we believe in severe models that replicate the common human phenotype. And in this condition, the ones who are going to need gene therapy at first are severely affected, and they are the ones that have high levels of ammonia and may need a liver transplant. So that's where the focus has to be for animal models, and that's why we picked that one. We're also believers in benchmarking therapies to see how good one is, any one versus another in the same animal models. So we're working on this. It's not always embraced by everybody to do this because it can cause one thing to compete against another, and not everybody wants to do that. And obviously partnerships are very important, and I appreciate the invitation to speak here and to continue our partnership with PAF. And I want to close by expressing my deep gratitude to everybody that's been in our study, all the supporters, and everyone else, especially our students who I didn't have a chance to shout out. I had a lot of students in my lab last year. We had 5 kids graduate. 2 went on to get MD PhD scholarships. The other one went to Northwestern, 1 went to UNC, and 1 went to Dartmouth. So they're learning, how they're learning about medicine is about PA. So to me, that's super exciting. And I will stop there for questions. I think I have 20 seconds to take questions, so, if anybody has anything they want to ask me about. Anybody?
Yeah, I have a question.
Oh, wonderful.
Thinking about the zebrafish, you see that really to me is super interesting. Did you say that people with mutations in ACAD8 and the other one, ACAD8B, exist and they're healthy?
Yes, that's correct. I should, it's not fully proven yet, but those patients have been detected by newborn screen, and some have had some mild symptoms. It's a question if they all, but most of them don't have any symptoms. Yeah. So just to say that.
My real question is, in the paper you showed that either the double knockout or the triple knockout do better. Thinking about translation, do you think one would have to target both ACAD8 or ACAD8SB, or would you go one by one, or how do you think that would work out?
I don't know if that. No, I don't know if that's a planted question, but I didn't go over this. But in the paper, we have an in silico modeling study that looks at the promiscuity of the active sites of these enzymes. So I think the way to do this would be to fully inhibit one and use an inhibitor that has some inhibitory properties on another. And what is not known from this work that uses knockouts is if you did such an approach, say purely in the liver, would that help? Yeah. Right, so we're looking at whole body knockouts. So I do think it could be something that if you had the right models, you could go with a medicinal chemist. And other people are working on this. There's a research in the space, and find inhibitors that have that property that are mixed, that are not absolute for both, because as you saw from the double mutant, that produces lethality in the double mutant. Whether you could use that to modulate, then combine it with something upstream, and then you could have multiple small molecules to dial the pathway in, is speculation, but it's an idea.
Or you can titrate the dose.
Or tighter, right? Or tighter the dose, right? That's the easiest thing to do.
That's super interesting.
Yeah. Thank you. Anybody else? Oh, go ahead. Yeah. Yeah, it doesn't target all sources of propionate. And so I didn't review this, but in the paper, we actually looked at post-translational modifications, which are aberrant in MMA and PA, and showed that in those fish, they still form PTMs, suggesting that we didn't get enough of the propionate load in that animal model to stop the phenotype. So the other sources, we don't know where they come from, but there's other sources beyond valine and ILU. Now in a human, you could sterilize their gut to minimize propionate from the gut, that's 30%, and then modulate BCAAs with diet and then inhibit, and then you could have an effect where you could combine all those sources of metabolites. Then you might see a much greater effect if you could do that. Yeah, so the question is, just to rephrase it so everybody follows it, MMA patients have a much higher frequency of developing CKD at a younger age versus PA. Why is that? That's a rephrase of it. Certainly under the electron microscope, if you look at, again, I go on based on human studies, there is a megamitochondrial dysfunction syndrome in the proximal tubule of PA patients. It almost looks like aberrant mitophagy to me under the EM. MMA has that as well. And again, the mitophagocytic component of it hasn't been fully elucidated. It's suggested in a number of studies. So it could be that in MMA, we're seeing an injury syndrome that's slightly different mechanistically, but it's the proximal tubule. Because both conditions produce chronic kidney disease, CKD, and they both have reduced nephronic mass and tubulointerstitial inflammation. But you need to go to the electron microscope to really understand at the level of the cell what the difference is. And then beyond that, the next thing I would say someone should do that's only been done once to my knowledge is to do immunogold affinity electron microscopy with specific markers to see whether or not in the tubule that's damaged, does it have a cytochrome C oxidase deficiency? Is there a Parkin defect? So to understand the mechanism. But I think there's similarity in terms of what it is. It's CKD. It's a mitochondrial injury syndrome. But in terms of the details, there could be a difference. And the other thing I would say is, in MMA, there's also been a debate, and I was switching to MMA for those who, I know it's a PA topic, but this is important, whether the free acid itself, which is much higher in levels versus a PA patient with, say, 3-hydroxypropionate and methyl citrate, whether that has an effect in itself. And that's still debated. Right? So in other words, it's a similar mechanism, but in MMA, you could have this giant metabolite load that's also, you didn't account for that. That's a difference between the two fundamentally different physiologies. We still don't understand how free MMA is handled by the proximal tubule. So that would be my short answer. Right. Anybody else? Oh, another one. Look at these questions. This is great. Go ahead. So those are some of the. So the question is, when are you going to dose your first patient? Right? That's. I'm just going to simplify it. One never knows when you go through FDA what they're going to. FDA, by the way, is the federal drug agency. What they're going to come back and tell you. What you really want is to have an IND, that stands for Investigational New Drug. If you have an IND approval, then you have your clinical protocol ready to go. There's steps involved. Just because you have an IND doesn't mean you can have a protocol. You need to have a protocol and have it approved by an IRB, then edited, then it has to go through a regulatory pathway. So in an ideal world, hypothetical thought exercise, the day you get the IND, the next day you get your protocol approved, the next day you treat. Right? Will that ever happen? No. So we hope it's going to be shortly from the approval of the IND, which, by the way, for those who are not following this, when you want to do a new treatment, if you have an FDA-approved product, it has to go through FDA. And FDA has to say, I approve or disapprove. And if they disapprove, you have to go back and say, I want to get approved. What do I do next? So there's an exchange. So that's assuming FDA says yes to the IND. So it's hard to predict the timeline. Unfortunately, I can't give you a firm answer for those reasons. Yeah. Wow, we have a lot of questions. I'm not, just for the timekeepers there, I think we should continue the questions. Go ahead. Yeah, so just to summarize, the one question is, what's the prognosis and outcome for CKD? And then the second one is, what can you do to mitigate? In terms of the prognosis and outcome for CKD, and again, I know we're talking a lot about CKD for not everybody in the audience. That stands for chronic kidney disease. So chronic kidney disease in itself is heterogeneous. There's some forms of CKD that progress more rapidly than others. And there's a whole separate discussion that can be had about renal physiology and why that's the case. It looks like the CKD of PA is slow to progress in terms of the bigger group of CKD patients. In terms of what you do to manage it, that's where I would say we don't have anything brand new and super insightful that's specific for PA. When a patient develops CKD with an organic acidemia, the approach is as you would for CKD in general. Blood pressure control, and then to consider a panoply of different medicines, and this has to be done in conjugation with nephrology. And unfortunately, that's where it stands. And I wish I had a better answer. What I can tell you that maybe is a tiny bit of hope is AAV9 vectors can transduce the kidney. And we can see this, but it's very low compared to the liver and heart. So unfortunately, right now, CKD is just a monitoring and aggressive symptom management problem for the patients without anything novel that I know of. I'm open to all ideas, and this is also part of why I think looking at new small molecules, that could be something to consider. So that's the first part of it. The second part is, yeah, we've been involved, not myself, in my group. Dr. Manoli really has worked with the Genetic Metabolic Dietitians International, the GDMI, that's what that stands for, to work on guidelines for PA dietary management. And when someone has CKD, I think one of the things, just as an overall principle, not everyone has, I should also say to the families, not all the kids that with PA develop CKD. Not everyone gets it. We don't know why that's the case either. I do think when someone starts to develop CKD, there has to be a real tight assessment of diet. And one of the things I think hasn't been done enough that I wonder about, based on experiences with MMA, again, MMA patients have an aggressive form of CKD, more aggressive than PA, is whether or not we should be doing antibiotic management more aggressively to try to reduce metabolite loads. Now, I think it has side effects, and it's not for everybody. And it comes with the risk of all the things from antibiotics. But I have evaluated patients with MMA that have had long-standing high-level exposure to antibiotics. And whether or not it helped their kidneys is uncertain in terms of preventing CKD, but whether it affected the rate of decline, which is what the metric one must use when you try to treat CKD. It's not necessarily about stopping it. It's stopping the rate of loss of GFR. So whether that's something else to think of is, again, a comment I would add. But, unfortunately for you, the bottom line answer is we don't have anything brand new, and, PA management approach with potential some additions per patient, that's what we would do. Yeah, so just to just say, this is something that's used to control ammonia levels, carglumic acid. I don't have anything new. I haven't studied it. I don't know who's taking it. And, if anybody has information to share about it, that would be of interest. Not everyone takes it. Some people have taken it and they think that helps them. Other people have said they're not as certain. And some people take it only when they get sick. And some people don't take it at all. And in some countries, to people that are listening online, they can't access it. And they take benzoate. And some families in other countries that take mainly benzoate, some of the families think it helps the patient. So I'm agnostic. Is it helping your family? Okay. Well, that's an amazing testimony, and it's just terrifying to hear that. I'm glad your daughter's doing well. To finish the comment, the old school treatment is sodium benzoate. And again, that it has its own set of symptoms and problems, and whether or not it does anything is a separate discussion. There's more to say about this related to glutamine metabolism, which I can talk to you about later. Yeah?
A big thank you to Dr. Venditti.
All right. Okay.